So it is my pleasure to introduce today's speaker, my friend, Stryder Meadows. Stryder and I went to graduate school together at University of Arizona, where he worked in Paul Krieg's lab on vascular development. And then we both did our postdoctoral work at University Texas Southwestern Medical Center, where he worked in Ondine Cleaver's lab and did important work on vascular development and dorsal aorta fusion. And then in 2014, started his own group at Tulane University in the Department of Cell and Molecular Biology, and where he has really been doing cutting edge research on HHT and focusing on SMAD4 mutant animals and characterizing HHT. And I think that one of the really cool things that Stryder has found that he's going to talk about today is that Angiopoietin-2 is a therapeutic target in HHT. And I think that his work is really relevant to lymphatic malformations, specifically like Kaposiform lymphangiomatosis, where we do see an increase in Angiopoietin-2 in patients, and it can function as a diagnostic biomarker for patients. So I'm really excited to hear about Stryder's work on HHT. And with that, Stryder, I'll let you take over. Oh, and just another housekeeping thing, actually, before I give it away to Stryder, is that if at any time during the presentation you have a question, please feel free to unmute yourself and ask your question, or alternatively, if you want to ask your question at the end of the presentation, just again, feel free to unmute yourself and ask. With that, I'll let you take it away, Stryder.
Yeah, and yeah, feel free absolutely to interrupt me at any point. Thanks, Mike. It's always nice getting together, and I appreciate the introduction. And also, I'd like to thank LGDA for the invitation to allow me to speak about the work that we've been doing in the lab. And sorry about the long title here, but essentially what my lab is interested in is understanding the molecular mechanisms that are involved in the pathogenesis of vascular malformations that are associated with this disease, hereditary hemorrhagic telangiectasia. And really what that's led us to do, as what Mike has talked about, is looking at and focusing on angiopoietin-2 and signaling via angiopoietin-2. And just to sort of set all this up, I have a picture here of one of our HHT animal models, and you see all the vessels here in blue. But what you have here is a large artery and a vein, and they're sort of entangled and commingling in a way that's similar to what you might see in what we call arteriovenous malformations in HHT patients. And so that's what I'm going to talk about today. Okay, so what is hereditary hemorrhagic telangiectasia, or I'll call it HHT? It's an autosomal dominant genetic disorder that's really affecting the endothelial cells, the lining of your blood vessels. And it affects about 1 in 5,000 people, so it's quite a lot of individuals. And so it's on par or as prevalent as other diseases such as cystic fibrosis or muscular dystrophy. And there's really two sort of main clinical symptoms that these patients present with. And one of those are these things we call telangiectasias. And these are malformations that essentially like to form in these mucocutaneous tissues. So they like to form in the mouth, the nasal passages, the GI tract, and also in the skin. And so here's the images of some of these telangiectasias, which are these red dots that you can see in all these different areas of the body. And really what a telangiectasia is essentially an enlarged or a dilated vessel. And the problem with having telangiectasias is they can lead to consequences such as severe nosebleeding. So these patients get really bad nosebleeds, but they also get internal bleeding, especially from the GI. So they end up becoming anemic as well. So the quality of life for these patients is really diminished. Now, another clinical symptom that my lab is particularly really interested in, one we think is a bit more severe, is the formation of what we call arterial venous malformations, or AVMs. And so if you think about the circulation, the cardiovascular system, the circulation of the blood, the heart is pumping blood out through the arterial system at a very high rate, and that blood returns back through the venous system at a much slower rate. Now, the arteries and veins don't directly connect together, but what connects them is these small vascular networks, or these capillaries. And so that's where the tissues receive the oxygen and the nutrients from these capillary vessels. But the other way that these capillary vessels serve the system is that they allow that this high pressure blood flow to really slow down once it gets into the venous system, which isn't really built for this high load or this high pressure volume. But what happens in these AVMs, as you can see with this picture here, is that you get an enlargement of this capillary structures and a weakening of the vessels. And it's almost like a direct shunt between the arteries and the veins. And the problem is that these particular AVMs are prone to leakage or hemorrhaging. And if that happens, you can imagine what would happen is you can have aneurysm, stroke, or even death associated with these AVMs. And unfortunately, these AVMs have a propensity to form in the brain, the lung, and the liver. And these are areas you don't want them to, really wouldn't like them to form, well, other than not form at all. And so here shows a picture of an AVM in the brain. And so really for patients, the only sort of treatment for these AVMs is a process called embolization, where you essentially put things in the blood vessels to stave off blood flow in that region. But there's no proven therapeutic reagents that have been used to be able to treat AVMs. And so there's a real need for these types of molecules to be found, or drugs to be found. Now, from a research standpoint, what's nice about studying HHT is that we know that HHT is caused by disruptions in TGF-beta signaling, this particular signaling pathway. And these are what we call haploinsufficient mutations, so essentially loss of function mutations. So here's a diagram I made of the transforming growth factor beta, or TGF-beta signaling pathway. It's more complex than this, but just to give you an idea of the signaling that's going on. And so really what we think in the field is that it's really happening on this end where HHT is tied to. And that is looking at more on the BMP910 ligands. And those ligands are essentially interacting and stimulating ALK1 type 1 receptor, which works with the endoglyn co-receptor to phosphorylate these regulatory receptor SMADs. And those receptor SMADs are only able to then enter the nucleus and affect transcription when they're bound by a co-SMAD called SMAD4. And so it turns out when you look at these patients, it's probably closer to about 95% of all patients have mutations in either the endoglyn, the ALK1, and the SMAD4 gene, with very few having mutations in BMP910 and presenting with an HHT-like symptoms. So, and I should also say that the vast majority of mutations lie within the endoglyn and ALK1, which referred to as HHT1 or HHT2, probably 90%. And the other 2 to 4% come from SMAD4 mutations, which also presents with an additional syndrome called juvenile polyposis. Now, recent research has actually shown also that they think that some of the driving forces of these vascular malformations are due to what we call loss of heterozygosity. And that is, if you go and you look at some of these telangiectasias and you look at them, for instance, in endoglyn, you find that you have the germline endoglyn mutation, but that normal copy, which you normally have, has now become mutated as well. Okay, so looking at more of a dual effect genomic-wise as far as affecting both alleles of this gene. So when I sort of really got interested in the field, essentially, most of the work was really looking at the receptor-ligand interface. But I became interested in really trying to figure out what are the downstream drivers of this pathogenesis of the HHT symptoms. And so therefore, what we decided to do is take a more different approach and to look at SMAD4 specifically and look at the downstream effectors. So what we did is we used the inducible Krelox-P system, where essentially what we're going to do is to knock out a portion of the SMAD4 gene to make it inactive, and then look to see what happens. And the way that we're doing is that we're sort of recreating that loss of heterozygosity background, where we're essentially knocking out both alleles of the SMAD4. We're doing that specifically in the endothelial cells themselves, and we can do that inducibly, so we can decide when we want to do it by adding something called tamoxifen. And so what you'll have here is the mutants that are referred to are SMAD4 inducible endothelial cell knockout mice. And the first way that we decided to look at this was to use a really nice genetic or a really nice vascular model of the retina. And what we did is we decided that soon after the pups are born, we gave them tamoxifen so that we can induce deletion of SMAD4. And that 7 days after birth, we then look at this highly vascularized retina to see what we could understand about this process. Now, I'll just show here on the control side over here, the reason we like the retina to begin with is it's really a nice system and that it's very stereotypical in the way that the vessels are formed. So you get this alternating pattern of arteries versus veins. And if you look here in the inset, what's really nice is that these arteries and these veins are distinguishable both physically and molecularly. But also what we liked is it has this particular vascular bed where you have an artery with in between capillaries attached to the veins. That was something that we liked about it. So now when we knocked out SMAD4, this is essentially what we saw in the retina. And I'll point you to this inset over here. And that is what we found was these really large direct connections between arteries and veins, which were forming these very large shunts. And what we essentially deemed as some type of an AVM-like formation. And this was very consistent, forming routinely in these knockout mice. And the additional phenotype that we saw was not only this AVM-like formation, but was an enlargement of the vessels, both in the arteries and the veins themselves, but also the capillary networks. So really what this model provided was essentially two main phenotypes that we see in patients, seeing these AVM formations as well as a vessel enlargement. And I should say, previous work done in the Endoglyn and ALK1 retinal models had shown similar phenotypes as well. And this is just showing the different types of AVMs that you might see when you're looking at any of these different types of mutants, you know, this sort of tortuous vessel formation and these connections and vessel enlargement. Now, again, though, what we want to do is to get downstream and figure out what are the driving factors involved with this. And so to do that, we took more of a genomics approach. And that is, we went in and we isolated the retinal endothelial cells from the control and mutant mice and did RNA sequencing to find out which genes were differentially regulated. And on top of that, which what we liked about SMAD4 being a transcription factor, is we did chip sequencing experiments. This was all done in vitro, looking at mouse endothelial cells. But what we've done is we've stimulated them with BMP910 to put them on that sort of side of the HHT pathway of TGF-beta signaling, and ask essentially where is SMAD4 binding throughout the genome, because that'll give us indications of where it potentially is regulating and what genes it's regulating. So for each of those, we got 1,900 to about 2,000 genes, which is a lot. So we simply integrated the two data sets to find out, or to bear out what might be the most important to us. And when we did that, we had this list of 212 genes. And really the story comes to us, what we got interested in is when we found Angiopoietin-2 and TEC, which is also referred to as TIE2, but I call it TEC. And what we found in these samples, and which is a sort of a theme in this talk, is that we had robust elevations of Angiopoietin-2 and decreased levels of TEC. And the reason we got interested in this is because these two are components of an angiopoietin-TEC signaling pathway. And I'm just going to spend just a little bit of time going on this pathway and a very simple, simplistic view of how the signaling occurs. And that is, you have your endothelial cell. And so this TEC is a tyrosine receptor kinase, so it becomes phosphorylated and signals downstream. And TEC is primarily expressed just in the endothelial cells. And it has two different types of ligands. It has angiopoietin-1 and angiopoietin-2. Angiopoietin-1 is thought to be primarily expressed on surrounding vascular cells like pericytes. And that interaction of angiopoietin-1 with TEC stimulates the activity of TEC, and for the most part leads to a very stabilized vasculature. But it's a bit different when you talk about angiopoietin-2. So angiopoietin-2 is the antagonistic ligand, and it's expressed on a subset of endothelial cells. But when it goes through this part of the pathway, it inhibits signaling through TEC, and it creates a much more active vasculature, which is what we think is happening in HHT, which is a much more dynamic or overactive vasculature. So thinking about this, what we see is increased levels of angiopoietin-2, which is going to inhibit signaling of TEC. We also have reduced levels of TEC in these endothelial cells, suggesting that you essentially have a net reduction in TEC signaling. And the other reason we got interested in this pathway is for a couple of reasons. One is that TEC signaling has been shown to be involved in regulating diameters or the calibers of the vessels. Interestingly, TEC mutations in patients where you lead to overactivation, too much signaling in TEC, which is opposite of what we see, actually leads to venous malformations. And then what we think was also really important was that looking at sporadic brain AVMs, so these are ones that not tied to HHT, showed that they also had elevated levels of Ange2 and decreased levels of TEC expression. So right here, I'm summarizing some of our published work to give you an idea of what we sort of came to when we started looking into these studies. And this is essentially the model, and I'll talk a little bit about this. But we have all the signaling going from the BMP9 to the TGF-beta signaling pathway, where essentially SMAD4 appears to be acting as a transcriptional repressor for Angiopoietin-2. So it represses its signaling and actually binds it at several different areas before the angiopoietin promoter. And those areas are actually conserved between mice and humans, suggesting it's there for a particular reason. And through this, you get a normalization or a normal development of the size of the vessels, the size of the endothelial cells, and which leads to a normal vascular bed. However, in our SMAD4 knockout mice, when you get rid of SMAD4, you essentially relieve that repression. And now what we see is we get a robust expression, robust amounts of angiopoietin-2 that's being expressed in the endothelial cells. That leads to increased vessel diameters, as well as increased endothelial sizes themselves. But subsequently, it leads to what we think it leads to is the formation of these AVMs as well. And so what we also showed is if you can inhibit angiopoietin-2, you might be able to go back the other direction. And so what we were fortunate enough is we reached out to Roche, which has made an angiopoietin-2 mouse monoclonal antibody, with the idea if we could use that antibody to functionally block and inhibit this excess angiopoietin-2, might there be a therapeutic benefit from using that? And here I'll just show you a little bit of that data. And to make it a little bit easier, let's just look at panels C and D. And so in C, what you have is our mutant that we've seen before. We get these large AVMs to form and these increased vessel diameters compared to our controls. Now, when we give these LC10, this Ange2 inhibitor, remarkably to us, we were quite surprised at how well it worked. These retinal, the vasculature of the retina looked really quite normal, And it turns out we could either inject or inhibit Ange2 before the AVMs formed and completely prevent AVM formation and these vessel enlargements, and they'd look like this. And we could also give this drug after the AVMs have formed, and you see a regression of the AVM formation and vessel enlargement. So this is really cool to us, because we are going from understanding what's happening in the cell and dictating to us, essentially trying to find molecular targets, and then be able to move that and to see if we can actually test that in our mouse model, which is what we've done here. Now, this work is published, and so the rest is unpublished, but really want to tell you about the work we've been doing to sort of push the story forward. And sort of that starts with looking at how we can expand this in the sense that of two senses. One is that most patients don't get retinal AVMs, but we know they get them in the lung, the liver, and the brain. Can we expand this to look at more clinically, physiologically relevant organs? And what we decided to do was actually to start looking at the brain. And we chose the brain for a couple of reasons. One is because you think of, as far as surgical remedies or even particular drug treatments, that the brain might be a harder area for access to be able to do these types of things. But the other thing, which I talked about earlier, that really got us sort of into it, was that looking at the data, looking at sporadic brain AVMs, showing the same relationship where we have too much Ange2 and little TEC. So that's why we decided to look at the brain. And there's more evidence now of this, and I'll talk a little bit about that later. Well, the second one was knowing that this all goes through the same pathway. Is Ange2 inhibition, could it be a universal sort of therapeutic approach, right? And the reason I present it that way is because we know, looking at the animal models, there are differences in the phenotypes, but we also know when we look at patients, there's different incidences of organ-specific AVMs. So for instance, lung and brain AVMs are more common than HHT1 than HHT2. So the question is, would this be universal type of therapy? And so really what we've sort of looked and tried to do is to understand what's the relationship with TEC and Ange2, also in the ALK1 and Endoglyn mouse models, and could inhibition of Ange2 have the same sort of therapeutic benefits. And the other thing I should say is that really there's no studies that have put all three models together, and we think that would be a powerful way to really look at this. And so that's what we've decided to do. So I'm going to show you the brains of all three different models, but first I'm going to describe to you how we look at them. And so what we do is something called a blue latex dye injection, where you essentially inject this dye into the left ventricle of the heart. It gets pumped out through this arterial system, but that dye is such a size that it can't get through the capillary beds, and it's absent in the venous system. However, you can imagine though, if you've got large capillaries or an AVM, then that dye is able to leak through, and that's what we're looking for. So let's look at some of these, the brains of these mouse models. And just to show here, the way we've set this up is for SMAD4 and Endoglyn, we've induced deletion into the same type of series, and we're collecting brains at 7 days after birth. The ALK1 model is a little different. These mice can survive for up to 48 and a little bit longer than that, 48 hours. So we had to tailor this a little bit different, where we induce deletion later and collect slightly earlier to get sort of these robust malformations to occur. So if you look on this left side, I'm just showing in this case the control for SMAD4, but essentially ALK1 and Endoglyn controls are the same. But what you see is essentially blue latex dye in the arterial system. And you can even see here, you look on the side view, you can see right here, there's actually a big vein, and all you see is blood, but you don't see any dye. So this is really hitting just the arterial system. And obviously, when you look at both the SMAD4, ALK1, and Endoglyn mutants, they're quite different. And it's really obvious to see that there's some vascular abnormalities going on. And in the next slide, I'll pinpoint a couple of those things. And in many cases, they're similar. In some cases, they're dissimilar, but I'm really more focusing on the similar aspects at this point. But to come back to looking at the lateral views, again, you can see these large artery and veins and the sort of intertwining of them in an AVM-like structure. Also can see this in the ALK1 HHT mutants and the Endoglyn ones as well. Now, let's take a look at some of the more specific phenotypes. And so one of the things that we decided to do as sort of a readout of AVM formation is to see how often we saw this dye in the veins, because the only way that it could get there is through some kind of a connection or enlargement of vessels. So you can see here, you have a main artery here on the ventral side of the brain. You see that same one in the stark blue, but then you see here in that lighter blue, essentially these giant veins that you don't see in here. Same thing with the ALK1 mutants. You see the veins that are expressing this dye as well. And with Endoglyn, you also see these veins expressing dye as well. So if you look at this right here, it's statistically significant as you see lots of perfused veins with the dye, suggesting that there's a lot of connections between these arteries and veins, or these AVM-like structures. In addition to that, we also looked at the diameters of vessels. In some cases, it's really obvious to see, but we focused on the basilar artery here. And here you can see from here in the SMAD4 mutants, the diameter is greatly increased. Same with the Endoglyn, the diameter has greatly increased, and not just that, but in other vessels, but this is our measurement. Now you'll notice though in the ALK1, you don't see, here it looks like an enlargement, but when you look at there's really not an enlargement in vessels. And I should say that's similar to previous reports looking at other vascular beds of ALK1, where there's not much of enlargement of vessels, although there are AVM formations. And you can also see highlighting the addition of the blue dye in the veins, where you don't see that in the controls. In fact, you can see blood filling the veins, but not dye. So again, these are coming back to sort of the two main things we're looking at: AVM-like formation and vessel enlargement seem to be apparent in all three models. Now, the other thing we want to do is then see if the same Ange-TEC relationship occurred. So the first thing we did was we isolated the endothelial cells from the brains from each different model. And in each case, what you see is you get a robust increased transcription of Ange2 and a reduction in TEC. And if you even take the proteins and ask the protein levels, you see the same trend or the same exact thing, where you have robust levels of Ange2 being expressed and reduced levels of TEC. And just as another way to show it, looking at sections of these brains, we're using in situ hybridization to essentially look at the RNA for Ange2. You can see in controls that Ange2 actually really isn't even expressed in the endothelial cells at all. It's expressed in the thalamic region and parts of the brain. But I think, as you can clearly see here, that Angiopoietin-2 gets robustly expressed throughout a lot of the blood vessels through both of those. And it wasn't just increased Ange2 and decreased TEC, but we also asked what's happening with the activity of TEC. So if you remember, I told you before, but increased phosphorylation equates to increased activity of TEC signaling. So you see here in our control mice, we have phosphorylated TEC, but when we look at our mutant samples here, you can see a reduction in the phosphorylated form of TEC, suggesting again that we really are having a reduced TEC activity in the brain endothelial cells that we're looking at. Okay. Now, the other thing that we really want to do, and to provide more of a comprehensive model of this, because no one's really looked at all three models, is we then went back and used sort of a genomic assay, and that is to do RNA sequence analyses and to compare that between all different mutations. So you can see here, when you do this, we have about 1,200 genes that are common between those. And if you then take those genes and do a gene ontology analysis, essentially asking of those genes, what type of biological processes are they involved in, you get a whole cohort of these readouts. But the only one that seemed to be common between all three of them was angiogenesis. And I bring that up because it's already been thought that HHT and the development of vascular malformations are due to an abnormal sort of reactive angiogenic process. But we would also say that we also don't know what the molecular signature of that process, the angiogenic process, is. So 1,200 genes is still quite a lot to genes to kind of go through. So then we went back and took our SMAD4 chip sequencing data to really nail it down to direct targets and integrated that as well. And so now between all of them, we have about 193 common genes. And if you do the gene ontology analysis between all four data sets, you get something that looks like this. And again, the only one common between all four of them is angiogenesis. And that came down to about 23 genes. And here what I'm showing is the heat maps of those. And within that, what you can see is a number of those that are upregulated between all three data sets, those that are downregulated between all data sets, and then some of those that are sort of mixed in between. But one thing you'll see is, obviously, we have angiopoietin-2 up. We have a whole other set of genes that are pro-angiogenic in nature. And that, what we think, is starting to sort of whittle down and giving it more of a molecular signature of this type of angiogenic process that's occurring in HHT. Now, the other thing that we think is really neat is that there's a lot of data, or not a lot of data, there's two main publications that recently come out where they've done really fantastic experiments, where essentially what they've done is they've gone in and isolated the brain vasculature, two different studies, looking at adult humans, in one case, fetal human and brain endothelial cells, as well as a number of vascular malformations. So they're taking these endothelial cells and doing single cell seq analyses to see what's the properties of these endothelial cells themselves. And what they found, when you're looking at all of these different vascular malformations, such as AVMs, cavernomas, or a number that are tied to tumor formation, they all show an increase, a robust increase in expression of Ange2. In fact, that was one of the main takeaways from these papers. So that was really interesting to see correlated with our data. And the other interesting tidbit was that some of these, the data suggested that this process is more of a reactivation of a developmental angiogenesis. And that was marked by a number of the same types of genes, which I have asterisks on here as well. And I'll just highlight a couple of those, like apelin and ESM1. So apelin is one factor. It's also a ligand, much like angiopoietin-2, that's pro-angiogenic in nature. And so it was really neat to see these same sort of molecular signatures coming through to the human patients as well. Okay, so with that said, now the question is, if we inhibit angiopoietin-2, how does that work on these different HHT mouse models that we have? And so on the top here, we have our IgG controls, And then we have our controls that are injected with LC10. And for the rest of these, I'm sort of going to ignore them, but essentially what you have is there's no difference. You see blood perfused vessels just in the arterial system. So the rest I'll focus more down here. And that is over here, you have your SMAD4 mutants, and then you're going to have on this side SMAD4 mutants that have been given this inhibitor LC10, Ange2 inhibitor. And I should also say that in all the different models, we're giving the inhibitor early, so it's more of a looking at a prevention in the formation of these malformations. So you look over here, one thing you'll notice in the SMAD4 mutants is you get a leakiness of the vessels. So you get a lot of the dye that leaks out. So it makes it actually a little hard to see some of these things. But as we've seen before, you get these avium-like formation, and you start to see an enlargement of the vessel diameters, as well as veins that have the dye in it. Now, when you apply your treat with the Ange2 inhibitor, you get a vasculature that looks a little bit more like the controls than it does like the mutants. So one thing that we saw, for one, is you saw less. Now, this one is a mutant that worked very well, but you did see a little leakiness, but nothing to the extent that you saw here, although we haven't quantified that. The other thing that we saw is that we still saw blue latex dye in the veins as well. So there was no statistical difference between that. But if you actually were able to measure the veins themselves, the diameter of the veins appeared to be much smaller when you treated them. And then if you look at the BA, the basilar arteries, the diameters of those, which where they were increased, went right back to normal. So here's a case where we think we've seen some nice improvements by inhibiting Ange2. We're essentially preventing these vessel enlargements and having some of a reduction in the severity, not such severity, but in the veins themselves, and a vascular that looks much more improved. So what happens when we do this with the ALK1 mice? So again, I'm going to focus down here on the bottom here, the mutant mice. Again, as we've seen before, you have veins that are perfused with this dye, which normally you don't, both here and here. And when you look at the mutants that have been treated in here, what you see is you still occasionally see veins that have dye, but at much less of a frequency. So you can see right here is that you see a lot less of veins that are filled with dye, suggesting that perhaps the severity of the aviums themselves have been reduced with using angiopoietin-2 inhibition. And also, as I said before, if you look at these, the total vasculature looks more like a control brain than it does in these mutant ALK1 HHT mouse models. And the diameters of these did not change, and we didn't expect them to do because they didn't initially any in the mutants as well. All right, so then what happens with the Endoglyn mutants? So this was really pretty cool because what we saw looked to be a complete prevention of any of these phenotypes to form. So when you look down here in the Endoglyn mutant, you see right here this sort of avium-like structure with the vein closely associated with the artery. You also have veins which are not expressing, but are contained with this dye. And you see these increased blood vessel diameters. And when you look at those that were treated with LC10, they're completely normal. They look just like those of the controls. So you can see here on the side view, you have this large artery. You see no dye within the vein, but you can see it in the blood. So the vein's there, but it's not being filled with the dye. And as well, you don't see veins in these other typical areas that you would see in the mutants. And you see a reduction in the diameters back to a normal or prevented from forming and back at a normal level. So this was really nice seeing a nice effect or impact of the Ange2 inhibition on the Endoglyn model. Okay, so this was nice. It looks like a potential therapy that could help all three models, maybe at varying degrees, but one that could maybe universally work. And so we also took this a little bit of a step further and asked and essentially interrogated the endothelial cells from the controls, the mutants, and the mutants that were treated with angiopoietin-2 to see if we could get more at what's molecularly happening. And so what we have here is we've done RNA seq analysis. Again, these are from isolated brain endothelial cells. We have our control brain endothelial cells. Then we have our SMAD4 HHT mouse model. And then what we have with the second set is we've looked at what happens when you isolated endothelial cells from those with SMAD4 that are treated with the Ange2 inhibitor and compared to the actual SMAD4 HHT model itself. And so when you put these data sets together, what you get is about 189 genes that seem to be in common between all three of these different, between these two different experiments. Another way to look at that is to look at the Go analyses again. So when you looking at just the set one right here, a number of processes, but angiogenesis being the foremost. When you look at the set two right here, angiogenesis being the second process most representative. And then when you go in and take the 189 genes that were found in common and look at the geo analysis, again, angiogenesis is the top term that comes up. But I think what puts this in a little bit better perspective is when you take the same data and you put it in a directed acyclic graph. And essentially what this is doing is taking all your top geo terms and putting together and correlating them in a way if they're related. And so when you look at this and you take all these, you get a number of these biological processes that all come down to angiogenesis, which again, is not surprising. And the other one that pops up is a lot of these processes involved in cell migration. And I point that out because there is evidence that cell migration is involved in this process. And there's some very recent evidence from Ann Eichmann's showing the same thing. But essentially to this, what it was saying was to us was that both in the process of forming an ABM or the pathologies, cell migration and angiogenesis were involved. And furthermore, when you treat with Angiopoietin-2, you're also regulating cell migration and angiogenic processes. And to show this in another way, if you look at the heat diagrams of each of these data sets, there's one particular region that's really quite marked, and that is that you see here in the controls a reduction in the genes expression, a huge induction of those genes in the mutants, and then those mutants that are treated with Ange2 inhibitor go way back down. And so if you blow that region of it up, what you end up seeing are a bunch of genes that are involved. And again, this part isn't really surprising, but to actually see the genes themselves, and genes that are involved in angiogenesis and cell migration. So we think the Angiopoietin-2 is having an effect on these two processes to essentially, in this case, prevent the malformations that are forming. Now, I should say we've only done this in the SMAD4 model. We'd love to do this in the other models as well, but we only have so much of this drug to use. And one other thing I'll point out, I talked about apelin being one of these factors, pro-angiogenic factors like Ange2 that gets expressed way too much. Well, what you end up seeing is that its receptor, when you're treated with Angiopoietin-2, that receptor goes way down. So this might be a way in which that increased apelin is being mitigated by reducing the receptor that it could be signaling through. Okay, so again, what we liked about this was sort of the comprehensive nature of it, but also getting down to sort of defining what is the type of angiogenic vasculature that is related to HHT, and then how is that affected when we look at Ange2 inhibition as a therapeutic model for these three different HHT mouse models. All right, one of the last things I'll just kind of go over with is essentially you're wondering, or we're wondering, is Ange2 inhibitors, are they available? Could we use that? Could this be a type of therapy for patients? And well, there are a number of angiopoietin monoclonal antibodies that have in clinical trials. And most of those are designed looking at tumor formation or ocular diseases, ocular diseases involving the vasculature, such as wet AMD or diabetic macular edema. And in most cases, though, what's happening is those clinical trials are combining Ange2 inhibition with VEGF inhibition, okay? But one thing that's been really interesting is that Roche has made a bispecific antibody that will simultaneously target Ange2 and VEGF-A to inhibit them. And this has recently been FDA approved. And the experiments that they did were actually to show that it could be used to treat wet AMD, wet age-related macular degeneration, as well as diabetic macular edema. And what they found was that typical treatments are usually a VEGF-A inhibitor. But using this particular drug, you could go longer periods of time without getting injections and some improvements as well. So I'm wondering if this might be something that could be usable for HHT patients, which have to deal with bleeding, with a lot of other associated problems within these patients. Okay, so with that, I'd just like to summarize the work that we have done, and that is especially the unpublished part. We really taken a cross-model comparison of all the HHT models, but putting that into more physiologically relevant organs such as the brain. And we showed that all three of them have defects in the brain vasculature in these models, and some of them are similar in ways and different, but they all have AVMs, and they all have, well, they don't all have vessel enlargements, but two of the models have vessel enlargement. And we showed that the same robust elevation of Ange2, reduced TEC, is same throughout all three models. And we think that's also leading to reduced signaling in the endothelial cells through TEC. And we think we've identified a unique in the sense of not just HHT, but maybe unique and conserved angiogenic program that's in HHT and maybe other vascular malformations as well. And then if we then go in and look at Ange2 inhibition as a means, as a therapeutic remedy, test those into those models, we seem to have improvements depending on the model, but marked improvements. And those seem to be tied to effects on angiogenesis and cell migration processes. Okay, with that, I'd like to thank the people that really do the work. And first I'll start with Xingyan Zhao. She's the one that's really done all of the brain vasculature project with this, with a little bit of help of Jenna and undergrad Nikita. And then all the previous work that was done in the retina and really started everything was really talented graduate student Angela Christ. And I also like to thank all of our collaborators, which have helped give us these mice to be able to do these types of studies. And I'd like to thank Roche for actually giving us the drug. It's really enabled us to do a lot more than I thought possible. And just lastly, I'll thank those that fund our research. So thank you for your attention, and I'll take any questions.
Great job, Stryder. Beautiful slides and just very elegant work. And I'll get the conversation started. I was curious about the durability in response to the Ange2 inhibitors, and whether, if you get mice past this sort of window in early development, do you protect them against AVM formation later on in life?
Yeah, that's a great question. And it's not really one that we can answer. So, with the mouse models that we have, Endoglyn and SMAD4, once you induce deletion postnatally, they die within P8 to P10. So you don't really have a long time to do this. And ALK1 they die so quickly, it's a really tough experiment to do. But it's a really valid question, because the question is, is this a lifelong sort of therapy strategy, because it also prevent new AVMs that are forming or the progression of AVMs. There's not really a good model that's published yet that's really could address that, but that's something we would really much like to answer. Yeah.
Thank you. And Carrie, we'll let you ask your question.
Yeah, really, really nice talk. I like the project and stuff. I have a little bit that builds on that, but goes from the question of the model and how you've been using it. Mm-hmm. You've been doing the knockout during active angiogenesis, right? And correct. A lot of times you're saying there's one knockout allele and then you're inheriting a second mutation. Have you thought about trying to do your models where you have an established vasculature, maybe even in a heterozygous, and then doing a knockout of the other allele in the endothelium and see what happens? I mean, I think this is the question a lot of vascular malformations, right? Is how's the origin and how does it develop along the way?
Yeah, absolutely. That's a great point. So I'll say a couple things with that. One is, so there are people will look at, for instance, ALK1 heterozygote mice, and they'll develop really quite normally. They will occasionally form AVMs, but you can't, just alone, you don't really see formation of these malformations. What they tend to have to do is some kind of secondary insult to stimulate this. And what we've done is essentially provided the genetic insult right away to look at this. So there's definitely two different ways of looking at it. But yes, in our model, it's not really like a patient, right? So
Yeah.
one thing that we have done, which we're starting to work on, is something, and it kind of gets at your question a little bit, was we've modified our SMAD4 model, where essentially what our SMAD4 model is a floxed allele and then a knockout allele. So that's sort of like a human, where you've got a normal allele, which is going to be our floxed, and then the knockout. And then we can then induce and see what happens, right? And so what we see when we do that, and this is very beginnings work, is we really have to titrate how much tamoxifen we give, because if we give the same amount of tamoxifen, because they've already lost one allele, these AVMs get crazy and they get big. And so what we're actually doing is we're trying to titrate it down and work backwards to see sort of the composition of the AVMs as they're forming and progressing. Is can we make more of a mosaic model where some of the cells have that genetic mutation and see how AVMs develop, how many genetic mutations are needed to make an AVM, and look at those questions. But again, the problem becomes with these animals will die in a very short period of time. So I think it also comes to the issue of really starting to need more, see if there's additional models that can get you to more prolonged period that mimic more of a human. And I do know we have a collaborator that's starting to look in, and a strategy that you could use is to make human mutations in the mice, just like you would in a patient, to see if they develop these things in the same manner that you would see in a patient, that they're developing and progressing over time, although we think some of these are occurring early, but going through that.
Yeah, that would be an interesting question to see how fast they develop and when they start showing up, right? Your AVMs in that model.
Absolutely. Yeah, good question.
All right, Fayez, I'll let you ask your question.
Hi, Faiz Safari here from Neomed. Stryder, this is an outstanding work.
Thank you.
I have a question. You did show one of those array data that's notch signaling. It seems was highlighted somehow. And to my understanding, angiopoietin and notch or regulates notch. And I'm looking at it from a non-angiogenic, maybe for bias and selfishness purpose. I work on the skeletal system. Is there any changes, abnormalities, and any early developmental within the skeleton in those animals?
Ooh, that's not that I know of, skeletal wise. I mean, these things seem to be primarily expressed just in the endothelial cells, and it seems to be contained within that.
Right. And they regulate angiogenesis, which angiogenesis is very crucial for osteogenesis.
Yeah,
I'm just wondering.
True. I haven't come across anything that suggests that there's any problems with bone development or density or those types of things. But your link to the notch one is interesting, because it turns out if you overactivate or underactivate notch signaling, you can actually form AVMs. Now, and there's sort of a link between Ange2 and notch and these other things, and TGF-beta and notch. Although in the field, it doesn't look like notch is a central part of AVM formation as far as HHT is concerned. Although there's clearly animal models you can play with notch signaling, you can create AVMs, but it's not clear if there's a connection between those two.
Gotcha. I really appreciate it. One other question, if you don't mind. You did talk a lot about SMAD4.
Yeah.
And those mainly regulatory SMAD. I mean, is there any compensation of the inhibitory SMAD in these process?
Yeah, so this is something we really got interested in, and then we sort of had the data was sort of varying. We couldn't quite pinpoint it, and that's something that I was looking at. So for instance, there's SMAD6 and SMAD7 are inhibitory SMADs, and it's actually been shown, I think in the chicken, if you overexpress SMAD7, the vasculature forms these AVM-like structures. Now, when you look at the transcriptional data, we don't see much of a change with SMAD7, and then SMAD6 might change, but it's different between the models. So it's really hard to sort of understand, but we were really interested for a while, seeing if there was something with that to control the sort of signaling that's going through that, but that's still unclear to us.
Gotcha. Right.
Yeah.
Thank you.
Well, if there are no other questions, let's all thank Stryder. That was just an amazing presentation. And
Thank you for having me.
yes.
Appreciate it.
And bye, everyone. Thank you all for attending. Oh, one other thing, actually. The LMI, there's a funding announcement that I sent out, and we're accepting letters of intent for pilot applications. So those, we've sent 2 emails out about that, but we're going to send another reminder. The LOIs are due near the end of February, and you can find the instructions on the LMI website for how to submit your letter of intent. Thank you, everyone, and I look forward to seeing you at the next meeting.
Thank you, everyone.